Imagine you are facing a mysterious puzzle. Let's say a plant in your garden isn't growing like it should. It's not getting the right amount of sunlight, you think. But what if it's something more? Is it the soil, the water, or even the air around it? How do you figure out the cause and find a solution? This is where the scientific method comes in. It's the ultimate tool we use to solve problems, explore new ideas, and make groundbreaking discoveries. From curing diseases to understanding how planets move, the scientific method has been at the heart of every major scientific breakthrough. But what exactly is it? How do scientists use it to uncover the mysteries of life? In today's video, I'm going to walk you through the steps of the scientific method and show you how it's used in biology to ask questions, test ideas, and reach conclusions. By the end, you'll see how you can apply this method to your own life, whether you're solving problems or exploring the world around you. What is the scientific method? The scientific method is a structured approach that scientists use to explore and answer questions. It's based on observation, experimentation, and analysis. The word science comes from the Latin word scientia, which means knowledge. So, the scientific method is all about building knowledge through evidence. There are generally 6 key steps in the scientific method: observation, hypothesis, experimentation, data collection, analysis, conclusion. Let's go through these steps in detail and see how each one works in the context of biology. 1. Observation. It all starts with observation. The scientific method begins when a scientist notices something in the natural world that sparks curiosity. In biology, this could be anything from observing how plants grow in different conditions to noticing how certain animals behave in their environments. For example, let's say you are observing a group of birds in a park. You might notice that the birds seem to be attracted to certain types of food over others. This observation might lead you to ask, what makes these birds prefer some foods over others? Tip: when making an observation, ask yourself, what do I see? What stands out? What might this mean? 2. Hypothesis. Once you've made an observation, it's time to form a hypothesis. A hypothesis is an educated guess or a prediction about what might be happening based on the observation. In biology, a good hypothesis often explains why or how something happens. It's important to remember that a hypothesis is testable. This means it can be supported or refuted through experimentation. For our bird example, a hypothesis could be if the birds are attracted to the sweet foods, then they are more likely to choose fruits over seeds. The key here is to remember that a hypothesis is not a final answer. It's a starting point for further investigation. Tip: when crafting your hypothesis, use an if-then statement. This makes it easier to design experiments and predict outcomes. 3. Experimentation. Now that you have your hypothesis, it's time to design an experiment to test it. In biology, experiments help you control variables and isolate the cause of the observed phenomenon. For example, you could set up an experiment where you present the birds with 2 types of food, fruit and seeds, and measure which one they choose more frequently. There are a few important things to remember when designing your experiment. Control group. This is the group that doesn't receive the experimental treatment, and it helps you compare the effects of your experiment. Experimental group. This is the group that receives the treatment you are testing. Variables. You should control all other factors except for the one you are testing. In our case, the only difference between the 2 food options should be the type of food. Tip: in an experiment, Always keep track of what you change, independent variable, and what you measure, dependent variable. This keeps your results clear. 4. Data collection. During the experiment, you will collect data. In biology, this data can come in many forms, such as measurements, observations, or counts. For example, you might measure how much time the birds spend near each type of food or count how many birds choose fruit over seeds. It's important to collect data consistently and accurately. Using tools like rulers, thermometers, or stopwatches can help you gather precise data. Tip: data collection is critical. Make sure your methods are standardized so you can compare your results properly. 5. Analysis. Once the experiment is complete, it's time to analyze the data. In biology, data analysis often involves comparing the results between the control group and the experimental group to see if there is a significant difference. For example, if the birds spent significantly more time near the fruit than the seeds, this might suggest that the birds do prefer sweet foods. The analysis might involve creating graphs or charts to visualize the results, which makes it easier to interpret patterns and draw conclusions. Tip: when analyzing data, always ask yourself, is this difference meaningful? What does the data tell me about my hypothesis? 6. Conclusion. Based on your analysis, you will draw a conclusion. This is where you decide whether your hypothesis was supported or refuted. If the birds preferred the fruit, your hypothesis would be supported. If they didn't, you might need to revise your hypothesis and test it again. However, in science, a refuted hypothesis doesn't mean failure. It's just part of the process. Sometimes refuting a hypothesis leads to even more interesting discoveries and new questions to explore. Tip: keep an open mind. Conclusions might lead to new questions and ideas, and that's the beauty of science. The role of the scientific method in biology. In biology, the scientific method helps us answer a wide variety of questions, from understanding how diseases spread to figuring out how plants convert sunlight into energy. The scientific method is a powerful tool that drives scientific progress. Quick recap. To wrap up, here's a quick summary of the scientific method. Observation. Notice something interesting. Hypothesis. Make an educated guess about what might be happening. Experimentation. Test your hypothesis in a controlled environment. Data collection. Gather measurable data from your experiment. Analysis. Examine the data to see if there's a meaningful difference. Conclusion. Decide whether your hypothesis was supported or refuted. Question of the day. What is the first step in the scientific method? Only a genius can answer in the comments below. If you would like to watch my other lectures on topics like the lungs, kidneys, liver, brain and eyes, visit my channel to view my other videos.